Respected Vice Chancellor Sir, Professor K. Sitaram Rao Garu, Registrar AVN Reddy Garu, Academic Director Ganta Chakrapani Garu, Dean Faculty of Science Pushpa Chakrapani Garu, today's special guest Datri Garu, and all directors and Dean faculty members and dear students and participants. A warm welcome to all for this program. I am happy to share with you that the Department of Zoology has initiated the webinar series, and today is the 5th webinar organized by Department of Zoology. So I am very grateful to Datri Garu for accepting to give a lecture for this webinar. I hope this webinar would be helpful to all. I thank each and everyone for attending this program. And Datri Garu, Madam done a research in Usmania University and published national and international journals and have a great research experience. And I will now hand over the session to Chief Guest Datri Garu to deliver the lecture on this webinar.
Yeah, thank you, Kishore sir, for the introduction. So, good morning, one and all. So, thank you for giving me this opportunity. So, now I will share the PPT. Yeah, is my PPT visible?
Yes, visible, ma'am. You can continue.
Okay, thank you, sir. So today I'm going to discuss about the immune system in pregnancy. So it's a
Datri Garu, I think your voice is not audible. Hello.
Maybe internet issue. Check your internet first. Hello?
Yeah, I think some internal issue is there. Dear students, please wait few minutes. Madam. So dear participants, due to some internal issue, Madam disconnected and she will going to log in again. Please wait, all of you. Sorry for this inconvenience due to technical issue. Thank you for your patience. And all of you, I am happy to share with you. These webinars also recording and available in YouTube Live. So you can log in YouTube channel, our Dr. B.R. Ambedkar Open University YouTube channel, and you can listen different classes along with these webinars also. So you can utilize that, and whenever you are going to learn something and whenever you are reading our chapters, you can go through that videos also. Recording videos also are available, YouTube classes. You can go through that.
Yeah, I'm really sorry. There might be some technical issue. Yeah.
Oh, okay, madam. You can continue.
Okay. So pregnancy is a complex physiological process because during pregnancy, the fetus or the embryo, which is growing inside the womb of the mother, is having the genes from both father as well as mother. So that means it is recognized as a semi-allograft by the mother's immune system. So because it is half foreign. So therefore, successful pregnancy is a challenge to the maternal immune system. So there is definitely a chance that mother's immune system may react to the embryo and it may reject. But it is not happening in most of the pregnancies. So this is because during pregnancy, the immune system of the mother is getting suppressed by certain molecules of the maternal immune system. So this is called as microchimerism. That means the immune cells of both baby, fetus, as well as the mother, they are in continuous contact with each other. However, they are not activating each other. Okay, they are in like collaboration with each other. So this is a well-organized occurrence during human pregnancy. And these fetal cells, they usually enter the blood of the mother and they will induce the activation of the maternal immune system. But still, it is not getting rejected. So here, the interaction between the mother's immune cells and baby's immune cells is having, the interaction is going at the place called decidua. So decidua is nothing but the mucous membrane which is present in the pregnant uterus. So usually we know that there is a layer called endometrium. So it is a layer which is it is a lining inside the uterus. So usually it is shed like every month during the menstrual cycle whenever the egg is not implanted. But during pregnancy, as the fertilized egg is implanted and it undergoes growth division. So during that point of time, this endometrium, it will get modified to form the So this immune modulation was explained in 1953 by Medaver. He was his scientist, so who actually explained this concept of fetal allograft.
System line, photo like
Sir, can you unmute? Can you mute? So Medaver is a scientist. He actually first explained the concept of fetal allograft. So how the immune regulation is happening between the mother and the fetus. So what he explained was there are three possible ways that this regulation is happening. So first one is the anatomical separation between the mother and the fetus. That means the mother and the fetus are not directly in interaction with each other. So there is a separation, anatomical separation. That means there are some layers which are physically separating this. Sorry, is my voice audible? Okay, thank you. So, therefore, because of this anatomical separation, there is a regulation of maternal immune system is happening. Then the reduced antigenic property of the fetus. So though it is antigenic in nature, so in the immunology, if you see antigen, antibody, so which one is called as an antigen? Antigen is nothing but which can induce the reaction in the immune system. So some antigens are very strong, some have weak interaction. So although fetus is inducing the immune response in the mother, but it is weak interaction. So therefore, rejection is not always possible. And the third one is the immunological inertness of the maternal immune system. That means though the mother's immune system is getting activated, still there are some molecules which are suppressed and which will try to continue the pregnancy. So here, the fetal cells are separated from the maternal immune cells at extravillous trophoblast. Okay, so this is nothing but the trophoblast is the layer in the like fetus, growing fetus, right? So in here in the trophoblast, actually some cells which will be suppressing the maternal immune system. So here, if you see, this is the uterus and this is the placenta. So here, these at the uterus, we have natural killer cells. This is of maternal origin. Okay. So we have macrophages, regulatory T cells, hormones, cytokines, chemokines. So there are various aspects from the mother side which are interacting with the baby. Whereas from the fetal side, that is placenta, we have some molecules like HLA, human leukocyte antigens, then some cytokines, chemokines, prostaglandins, hormones, etc., which will be interacting. So these two aspects are in continuous interaction with each other to establish successful pregnancy. So around 50% of the cells in the decidua are of maternal immune cells. So that means, see here, this orange color one is the decidua. So we have a lot of immune cells. Like immune cells, we have T cells, B cells, natural killer cells, dendritic cells, macrophages. So all these will be getting activated by the fetal cells, trophoblast cells from the fetus side. So here my hypothesis is that, so whenever the maternal immune system is suppressed, it will continue the pregnancy. But suppose due to any problem, if the maternal immune system is not getting suppressed, so then in such case, there is a chance of rejection of the fetus. That means abortion can happen. So usually if there is any problem, it can happen multiple times. So therefore, it can result in various pregnancy-related disorders such as recurrent pregnancy loss, preeclampsia. Preeclampsia is nothing but it is associated with the hypertension. So any of these things can happen. So why this rejection or why this breakdown of immune system can happen? So this is because of polymorphisms of immunoregulatory genes or inflammation due to cytokine shift. I will explain what are they. And majorly, the molecules which will suppress the maternal system are major histocompatibility complex, so which are present in the extravillous trophoblast cells. That is HLA-G. MHC complex, nothing but human leukocyte antigens. So these HLAs which are present in the trophoblast cells are the primary immunoregulatory cells. That means they will actually suppress the maternal immune system. They will directly come in contact with the mother's immune cells. Okay, this is the fetal origin. So the HLA from fetus is coming in contact with the mother's immune cells, and they are trying to suppress the maternal immune cells. So this is how actually the suppression takes place. Suppose if this suppression is not taking place, then it can result in a disorder that is called recurrent pregnancy loss. So my area of interest is recurrent pregnancy loss. So it is defined as 2 or more consecutive losses of fetal product before 20 weeks of gestation. So around 5th month, like before 5 months of pregnancy, like 20 weeks. So there will be loss of fetus. So this can happen multiple times, at least 2 or 3 times. If it happens, then we can consider it as a recurrent pregnancy loss. So usually once it may be possible for the normal woman also, healthy woman also. Sometimes it can happen, but if it is happening recurrently many times, then we have to be serious about it. So the prevalence of this condition in India in pregnant women is around 1 to 2%. So usually see the pregnancy loss can happen at any point of time during pregnancy. That means during implantation it can happen, then decidualization. There are various important stages, placentation. So during any point of time it can happen. And this recurrent pregnancy loss usually happens during implantation. So it is because of the implantation failure. That is before 5th month, early stages of the pregnancy. Then another aspect here is cytokine shift. So here, if we can just understand, you just imagine that during early stages of pregnancy, what happens? This embryo, first the egg is fertilized, and now this zygote goes and it has to implant. Okay, it will divide and then it goes and implant in the endometrium. So implantation is nothing but it is going and attaching to that area. So attachment is nothing but here it has to create a wound-like environment in the decidua. Okay, that is endometrium. So wound-like environment is pro-inflammatory stage. So first 3 months of pregnancy is usually, that is first trimester, it is pro-inflammatory phase. So it is like a wound-like condition. So that's why most of the women, they experience nausea during first 3 months. Then from 4th, 5th and 6th months, the second trimester, it will be anti-inflammatory. That means once it gets attached, now the next stage is it has to grow. It will start growing from that. Okay, when it starts growing, that is anti-inflammatory. Then again, after it reaches certain stage, again the last stage is parturition, that is delivery. So delivery is also like it has to come out. It is also like a wound-like situation. Again, it shifts to pro-inflammatory phase in the last trimester, 3rd trimester. So that means during pregnancy, the inflammatory stages has to shift from pro to anti, again to pro. So this is called a cytokine shift. So this has to happen naturally. It has to successfully shift from one phase to other. Suppose due to any cause, if it is not shifting, that means continuously it is in pro-inflammatory or continuously it is in anti-inflammatory stage, then in such cases, also it can lead to pregnancy loss. Okay. So this is how it happens. So during first trimester, it is inflammation. So how this inflammation is created, it is by the cytokines, pro-inflammatory cytokines, that is, they are secreted by helper T cells 1, subset 1. They secrete inflammatory cytokine. Then in second trimester, these helper T cells set 2, okay, they will secrete anti-inflammatory cytokines, and therefore this will create an anti-inflammatory stage, and it will allow the growth of the baby. Again, in the 3rd trimester, it leads to the pro-inflammatory stage. So now we came to know like what are the possible causes of recurrent pregnancy loss. Okay, one is immunological. All these aspects are immunological. That means either it can be due to inflammatory stage, shift failure, or it can be due to the loss of molecules which will suppress the maternal immune system. However, there can be other possible factors also, like anatomical. Some women, they have some anatomical defects in the uterus or fallopian tube or etc. So that could be a possible way. And then infection during pregnancy, there could be some infection. Then lifestyle factors, smoking or alcohol consumption, etc. And then ovarian reserves can be depleted, and that could be a possibility. Then immune system, then genetic. So various genes will be playing important role during pregnancy. And here, if it is genetical, definitely we will come to know it will run in the family. So it's a hereditary. So usually that is also a possible way of pregnancy loss. Then endocrine, like various hormones might play an important role. Then chromosomal. So a lot of chromosomal abnormalities can be seen during pregnancy. So, and it can be usually detected in the early stages of the pregnancy, and it can be if it is not curable, then usually the doctors suggest, they will counsel whether to go for the abortion or some treatments, etc. So all these are various factors of the recurrent pregnancy loss. But still, only 50% of the cases we actually know what is the reason. And the other 50% of the cases, RPL cases, we actually don't know what is the reason. Okay. So my hypothesis is, so if we take that 50% where we don't know actually what is the reason, so immunological factors may play important role. So here, as I discussed, HLA-G, human leukocyte antigen G. So we have various HLA-G molecules, HLA-A, B, C, okay, then E, G, F, A and D, Q, D, R, like that, we have many. So why I am discussing only about HLA-G here means, so HLA-G is very much restricted during pregnancy itself. That means it expresses only during pregnancy, and that too in the trophoblast cells. And it is rarely expressed in other places other than the pregnancy. Okay. And it is important molecule. It will regulate the expression of T cells, B cells, dendritic cells, then natural killer cells, endothelial cells, all these cells. So in this image, you can see it has, these cells have various receptors to which this HLA-G will go and bind, and it will suppress their function. So in this way, actually, this HLA-G is going, interacting with the mother's immune cell, and it is suppressing the maternal immune system. So this is a possible mechanism. Then another aspect here is, so in the IVF, in vitro fertilization, so usually when during implantation, they will check the levels of soluble HLA-G. Okay. So if the soluble HLA-G levels are good, then the implantation chances are more. So this, there are many studies which have actually conducted this type of experimentation. Okay, that means the role of soluble HLA-G. Soluble HLA-G is nothing but, so the HLA-G is of two types. One is membrane bound and soluble. Membrane bound is nothing but it is attached to the trophoblast cells on the cell membrane, whereas soluble means it is a secretory form. Okay, so this secretory form is important, even the membrane bound form. So both are important for the regulation of the maternal immune system. So here, this HLA-G has 7 isoforms, and it is located on the 6th chromosome, P-arm. It has 8 exons and 7 introns. And after alternate splicing, we have 7 isoforms, 4 are membrane bound and 3 are soluble. Okay, as I told you. And here HLA G1, G2, G3, G4 are membrane bound. G5, G6, and G7 are soluble. So here the full length isoforms are HLA G1 in membrane bound and G5 in the soluble. So they are more or less same, equivalent, but one is membrane bound and the other is soluble. Then comes 3' UTR and promoter. So this HLA-G, unlike other HLA molecules, they are not much polymorphic. So what is polymorphism? Polymorphism are nothing but changes in the base pairs. Okay, so every two organisms, like whether it is human beings or any other organisms, if in case of human beings, no two human beings are same. Okay, so although 99.99% they are same, but still there is some difference which is making them unique. So that difference is because of this polymorphism. So not all polymorphisms cause diseases because it is a natural phenomena, but some polymorphisms can cause various diseases. So in HLA-G, when compared to other HLA molecules, there are less polymorphisms. Okay. So, and if the polymorphisms are present also, they are present only in the two regions. One is promoter and other is 3' untranslated region. So promoter, you all know, like it is a starting portion. Okay. Then 3' UTR is towards the end. Okay. These two ends are very much critical and crucial for the expression of HLA-G. And apart from this regulation of immune system, the other important role of HLA-G is in the angiogenesis, that is blood formation. Okay, so because the blood vessels has to form for the new baby, right? So, and a lot of blood vessels will be formed because they have to grow. So therefore, remodeling of these spiral arteries is actually, it involves this HLA-G molecules. So based on all these facts, so I hypothesized in my PhD work that this 3' untranslated region, which is very much important for the expression of HLA-G, maybe if there are some polymorphisms in this 3' UTR of HLA-G, this HLA-G is not expressed properly, and therefore when HLA-G is not expressed properly, the maternal immune system is not suppressed. When mother's immune system is not suppressed, it will reject the baby, causing the abortion. So that might be the cause of recurrent abortions or recurrent pregnancy losses. So this is actually my hypothesis of my PhD work. So why 3' UTR means 3' UTR region is having various binding sites of microRNAs. So there are small RNAs, microRNAs are the small RNAs which are specific to or complementary to the mRNA. Okay, so microRNAs are present normally in our body, not associated with any disease. So what these microRNAs will do, they will go and bind to specific mRNAs and they will stop their expression, or they will break down that mRNA, target mRNA, and they will try to suppress the expression. Okay, so here as this 3' UTR region is having many binding sites of microRNAs, these microRNAs goes and they will cleave the mRNA of HLA-G, and therefore it is affecting the pregnancy. So that is what I am looking into, whether this is possible or not. So for this, actually, I have collected blood samples from the patients and controls. So control is nothing but any woman who have at least 2 children without any history of abortions. Okay, no induced abortion or spontaneous abortion or anything. So healthy children, healthy pregnancies. So they are considered as controls, whereas any woman who had 2 pregnancy losses at least earlier, so they were considered as patients. So then I have excluded them women who had only one spontaneous abortion or induced abortions or any known problems like anatomical problem, hormonal imbalance, etc. So I have excluded from the study. Then once I collected this blood samples, then I have isolated DNA from that blood samples and I have done PCR, polymerase chain reaction, which is specific using specific primers designed for amplification of 3' UTR region. So these are the forward and reverse primers, and the PCR conditions which I used for my PhD work are explained here. Then after I amplified the PCR products, then I have checked the PCR products using agarose gel electrophoresis, 2% agarose gel electrophoresis. Then once I confirmed that, okay, the region is amplified, I have given it for the Sanger sequencing. So before that, while collecting the blood sample, I have also prepared a questionnaire. Okay, so using the questionnaire, I collected various demographic, clinical, and family data. So what kind of questionnaire I have collected, I have designed means, so the risk factors that may be possible. So why the pregnancy loss may happen. So one is residential area. That means where the subject is staying, whether it is an urban area or rural area. Then educational background. So educated or uneducated, because they have to, they should have the idea like whether it is there is any hereditary problem or any health problems, etc. Then occupational status, non-workers and workers. Then economic status. Okay, so based on their income, like high income group, middle income group, and low income group. Then consanguinity. Consanguinity is nothing but the marriages between relatives. So that is also a possible risk factor for various genetic disorders. So then diet, vegetarian diet and non-vegetarian diet. The nutritional status. So good, moderate, and poor. The number of abortions. So once I got to know how many number of abortions a patient had, then I divided the group into less than 3, that is at least 2, and more than or equal to 3. And what is the nature of abortion? That is primary abortion, secondary abortion. Primary abortion means all their pregnancies resulted in the abortion. Whereas in the secondary abortion means, aborters means at least some pregnancies are successful. That means first pregnancy, they may get aborted, okay, abortion. Second pregnancy may be successful. Again, third pregnancy, abortion. So like that also is possible. That is secondary abortion. Then health complications, any previous health complications like BP, sugar, thyroid, etc. Then family history of abortions, unsocial habits like alcohol drinking, smoking, or any other gutka, pan, tambaku, or any other addictions. Okay, then hemoglobin levels, BMI, body mass index. So that is whether the person is obese or overweight or underweight. So all these I have collected. Then age at marriage. So whether it is very advanced age or very early age at marriage, and age at first conception also. Then menstrual cycle. So whether it is regular or irregular. So after collecting all these things, so actually I didn't find any much difference. So although there is a difference in some, means if we see the data, we may find some difference, but statistically, I didn't find any difference between patients and controls. Now, when come to the experimentation, so this is the gel image of genomic DNA. Like once I isolated the DNA, so this is the gel check I have done. And in the second image, after I have conducted the PCR, I could get this gel image. So where there is a polymorphism, which is called 14 base pair polymorphism in 3' UTR region. See, if we see, if there is a deletion in the deletion polymorphism is there, then I am getting only 127 base pair band. But if there is 14 base pair deletion, insertion polymorphism, then I am getting 141 base pairs band. So I collected random samples. So there I got the samples with deletion polymorphism and with insertion polymorphism. Finally, after completing my sequence, so I have selected all these polymorphisms. So total of 10 polymorphisms I have studied after sequencing. So in this 14 base pair polymorphism is associated with the stability. That means if based on the insertion and deletion, the alternate splicing and stability of mRNA is getting altered. Whereas all these other polymorphisms, they are associated with the microRNA binding. So after sequencing, this is how I got these are called electropherograms. So how we detect is, so this is, A to T, this is called insertion, and this insertion region is missing here. So instead of all these 14 base pairs, I am getting only G base instead of all these 14 bases. So here, if I see 2 peaks I am having. So this is nothing but the heterozygous. That means in the same site we have like 14 base pair insertion and deletion. So in the same way for the second polymorphism, so I have got C, CC, CG, 2 peaks, then GG. So these are the electropherograms of 5 polymorphisms, which I found variations. So I studied all the 10 polymorphisms, but out of 10, I could see the variations in these 5 polymorphisms. Like here at plus 2960 site, plus 3010 site, then 3027, 3035, and 3142. So all 5 sites, I could find the variation. And this is the allelic frequency. So how the frequency is observed in patients and controls. So suppose that means the deletion allele is present 47% in patients and 50%, around 50% in control. So like that, all the frequencies I have noted. Then out of these 5 polymorphisms, I got significance for only 4 polymorphisms. Okay, so all these 4 sites, I could find risk factors. That means here, the CC genotype is showing 3-fold risk, okay, towards the recurrent pregnancy loss. And here also in the second polymorphism, CC genotype is showing 6.66 folds risk. And in the third polymorphism, CC is showing 3-fold risk. And in the fourth polymorphism also, CC genotype is showing 3-fold risk. So that means in each polymorphism, the CC genotype is showing risk towards the recurrent pregnancy loss. That means women having this CC genotype, they produce low levels of HLA-G. Okay, so because of low levels of HLA-G, the maternal immune system cannot be suppressed, and therefore it results in the rejection of the fetus. That means it causes the abortion. So here the same thing I have kept it in the form of text. Okay. And here when I divided the patients as less than 3, that means the woman who experienced less than 3 pregnancy losses and more than 3 pregnancy losses, only one polymorphism, that is 14 base pair insertion deletion polymorphism, showed significance. So this insertion deletion, that genotype is showing protection. So that means deletion, you can see here, D means deletion. D allele, if a woman is having 2 copies of D allele, then she experiences abortion. There's a chance of experiencing abortion. Whereas the woman who is having insertion allele, she may be protected from the pregnancy loss. And if any woman who is having at least one insertion allele and one deletion allele, she will be protected from the pregnancy loss. Then I have measured the levels of soluble HLA-G based on this. So here I could find that in patients, low levels, here you can see in the controls, if I see 147 microgram, picogram per ml, if I see, then in patients it is only 58.93. So that means almost half, less than half, okay, of levels of soluble HLA-G was observed in the patients. So that means, I'm sorry. So this establishes that levels of soluble HLA-G are important for establishing the successful pregnancy. So even in the deletion, insertion deletion polymorphism, in deletion, you can see high levels, whereas insertion have low levels. So in the same way, I conducted, I'm sorry, my voice is not audible.
Is it audible, madam? You can continue.
Okay, fine. So, so for each polymorphism, I conducted the levels of soluble HLA-G, and it is correlated with the genotype, which I got, like whether it is risk or protection. That means the genotype which is showing risk. So in that patients, the levels were low. Okay. So finally, what I conclude from all these things is that because of the presence of that risk genotype, the levels of the soluble HLA-G are getting reduced. So because of that reduced soluble HLA-G, it is resulting in the abortion. And next, so what could be the potential effect? Like, okay, I'm telling like whether if C is present, it is causing abortion. If G is present, it is giving protection. So why C, what G? So what is the role? Okay, so in the first polymorphism, if we see, if deletion is present, then it is giving high producing allele, and in the insertion, it is associated with the binding of the microRNA 365B. So there are a lot of microRNAs which are playing important role. Then in the second polymorphism, C allele is showing affinity for some microRNAs and G allele is showing affinity for microRNA 5787. So like that, each allele is showing affinity for different microRNAs. So that's why actually we simply cannot conclude that, okay, because of binding of this microRNA, the levels are reduced. Sometimes in women having two alleles, that is heterozygous. So if both the alleles are showing high affinity for many microRNAs, even in that woman, reduced levels of soluble HLA-G can be observed. And the most established polymorphism is the last one, that is RS1063320, where G allele is showing high affinity for 3 microRNAs. So therefore, these microRNAs comes and binds to this 3' UTR region, that is mRNA, if G allele is present. Okay, and they will suppress the mRNA expression by degrading the mRNA. Whereas if C is present instead of G, then this much of degradation cannot be seen. So the levels will be moderately high when compared to the G allele. Okay. So these are the outcomes of my study. Then what could be like future aspects? Okay. Here, if we can make this, if we can actually do the same experimentation with a large number of samples, so if the studies are recommended, like studies are recommended with a large sample size, then we can actually make this HLA-G as a practical marker to predict whether the mother's immune system can tolerate the semi-allograft fetus or not. Okay, and as one half of the fetus is from the father HLA-G type and the other is from the mother's. Okay, So actually there is a treatment which is possible whenever the woman becomes pregnant. So actually it recognizes the father's HLA and they secrete antibodies. Okay. And these antibodies must be blocked by using blocking antibodies. And this can be done by using some injections. Blocking antibodies can be added. So in such cases, actually, we can prevent some miscarriages. So that is a possible therapeutic way. So this is how it can happen. So usually if normal blocking antibodies are present, they will usually block the antigens which are expressed by the baby. But if the blocking antibodies levels are low, then usually these antibodies will, the maternal immune system activating the antibodies, and that antibodies comes and rejects the baby. So we can actually inject these blocking antibodies and we can protect the fetus. And this blocking antibody levels can be measured by using flow cytometry. And other than this, there are other two treatments. One is like during this condition, white cell immunization to increase the blocking antibody levels, and IVIG, intravenous immunoglobulins. Okay. And the other is like giving immunosuppressive drugs like prednisone. So previously they used to give immune suppressive drugs, but now they are not actually giving that. So there are few treatment possibilities. If it is genetical, so it definitely requires some counseling. But if it is due to some any other spontaneous immune reaction, then some treatments like this can be applicable. So from this preliminary study, so what I like to conclude is that genetic screening of promoter as well as 3' UTR is necessary for establishing the role of HLA-G. So that means not only the single polymorphism, but haplotypes. Haplotypes is nothing but a combination of polymorphisms. If we can screen them and establish their role in pregnancy, then actually we can create a marker. Actually, we can make this HLA-G as a marker to predict the chances of implantation and chances of successful pregnancy, not only in vitro fertilization, but also in the normal pregnancies. So here, this polymorphism data along with the expression levels. So we have to estimate the levels of HLA-G also. So together we can establish the possible causes of recurrent pregnancy loss and also to develop certain treatment options. Okay, thank you. So if you have any questions, you can ask.
Madam, there are 7 doubts or questions in chat box. You may open and answer them.
Yeah, just a second. Sorry, can you read it out? Because I have like reconnected again, so I lost. Yeah.
These are not questions, you know, it's okay.
Okay.
Students, if you have any, one person raise your hand.
Hello. Hello.
Hello. Yeah, please.
Good morning. Madam, I have one doubt. Actually, early pregnancy is some have pregnancy is confirmed. No? So that somebody in early pregnancy, abortions are mostly will be.
Yeah, continue. Yeah, okay. Sir, your voice is not audible. Yeah. Hello?
Hello?
Yeah.
How?
Please go ahead. I can listen your voice.
Voice is not clear, but hello, sir.
madam.
Actually, I'm in school, madam, so I'm muted.
Okay. You can type in the chat box if possible.
Okay.
Hello.
Hello. I want to
madam,
mute, madam.
మనకి ప్రెగ్నెన్సీస్ కన్ఫర్మ్ అవుతుంది కదా, చాలా తక్కువ మందికి. అలాంటి టైంలో అబార్షన్స్ చాలా మందికి కన్ఫర్మ్ అవుతాయా? బ్లీడింగ్ అవ్వడం కానీ, అబార్షన్స్ అవ్వడం కానీ. హలో madam.
Let me read out this. In some pregnant women, vomiting sensation will continue till 8th to 9th month. What is the reason, madam? Can you hear?
Hello, madam.
Hello, can you read, hear, madam? Hello, Dhatri, madam, are you there? Hello, Dhatri, madam, are you there? Hello, you're not audible.
Hello, yeah. They have not unmuted my microphone. Yeah, now is it audible?
Yeah, it's audible. Did you read the chat box?
Yes.
She said that, why vomiting sensation will be there after 8th and 9th month. What is the reason, she said.
Okay. So
and
first
another one is, what is the function of membrane HLA-G?
Okay, sir. So first somebody asked why the size of cervix and all is small. So it's a genetical. Okay, it's an anatomical. It is nothing related to any health complication. Okay, so some may feel some health complication and others may not actually trigger any health problem. And the other is why some will experience nausea and vomiting. So here, as mother's immune system, so it is like if anything enters into our body, see, our immune system will react. Okay, whether it may not be abortion, but still a small amount of reaction will be there, because that baby is not from our own body, right? So it is something which is coming from outside and it is growing inside. So definitely our body, there will be a lot of hormonal changes. Okay, so that will be taking place. So a lot of changes will be, physiological changes will be taking in the mother's body. Okay, so because of that, and also I said like pro-inflammatory, anti-inflammatory shift. Okay, because of all these things, some women will experience nausea, mainly because of this cytokine shift and hormonal imbalances. Okay, so I hope I have cleared the doubt. And the other one is HLAG, and what precautions should we take if they clear? Okay, so it is all because of if we are not taking proper care, so that can cause. And of course, I am not a medical doctor, so I cannot tell you the precautions. So I can just only tell what is the possible reason. Okay. And what is the role of membrane-bound HLA-G? So here, so membrane-bound HLA-G also will interact directly with some cells. So because cells will be in the contact, okay, cell-cell communication is also possible. Okay, so that's why this membrane-bound HLA-G also interacts with the receptors on the other cells. Okay, so that is one possible, because we all know like cell signaling. Okay, so what happens, the membrane-bound molecules will send the signals to internal part of the cells, and they will make the changes in the cell. So in the same way, this membrane-bound HLA-G also will result in the immune suppression. And soluble form is nothing but it's a secretory form. It actually travels through the blood, and it will interact with the maternal immune cells, and it will suppress the mostly natural killer cells, uterine natural killer cells. Okay. Then some women early pregnancy. Okay. So yeah, see, abortion is quite possible. So that's why we like, even if the elders say or doctors say, whoever it is, at least 3 months we should be careful. So that means during the implantation process, so our like tissues and all these are very sensitive. So we have to give certain amount of rest, okay, certain amount of time to make the natural process happen. So that will go ahead and it has to implant. It will take some time. Our body will respond to it. So we have to give a proper time to establish the pregnancy. Okay, if we are making more stress, what happens? So as I told you, a lot of hormones, everything is involved in the successful pregnancy. They may get disturbed. Okay, if you are taking any stress, whether it is physical or mental. Okay, so what happens? The hormonal imbalance may happen, or our body may respond in any negative way, and it can abort. Okay, but recurrent pregnancy loss, it is a like a repeated process. So normal abortion is different. So if it is due to some accidental,
Excuse me.
yeah. Let me continue. Just I will complete this question. So every pregnancy, there is a chance of abortion. Okay, if you are not taking proper care. But that is a normal process. If we take proper care, it will get successful. Okay, but here in the recurrent abortion, though we take bed rest or whatever it is, still there is a chance of abortion. This is because of some genetic and immunological aspects. Okay. Yeah. So somebody is asking another question. Yes, are there any questions?
No, ma'am.
Okay. I hope I have cleared your doubts. Means only the take home message I want to give is, don't, like, we should not take the pregnancy as a take it granted. So most of the, like any woman, whether it is educated or uneducated, rural, urban, any background, like we don't know how many things are happening inside to establish that pregnancy. Okay, how much like compromises the body is making, how much suppression is happening inside. Okay, so there is a lot of regulation of various aspects which will be going inside the body to establish the pregnancy, successful pregnancy. Okay, so there is no problem. Like if we encounter any problem or any issue, we should go and talk to doctor, and we should make it clear there are treatments for all the things. Okay, all the problems. So nothing to feel shy. Okay, so we have to make use of the medical technology, and it will result in the successful pregnancy. Okay. Yeah. Yeah, that's what I'm telling. Like it can happen. Early stages are very like delicate, because anything can happen, because it is very like what you see is single cell, no, zygote. So it's very sensitive. So we have to give some time to like establish. Okay, so of course we even don't know, like early pregnancy means even until it gets confirmed, even the mother, she may not know. Okay, but there can be some underlying causes, or some unfortunately due to some accidental things it can happen. But if we are expecting, at least in our mind, we will know, like, okay, if we have any symptoms or anything, we should be careful. Okay. Yeah, any more doubts? Okay, if you don't have any doubts, so let me conclude.
Close transportation has been enabled. Cancel it.
Okay. Now, I thank you, Dhatri Madam Garu, for your elaborative and very useful lecture. And because of your lecture, students have raised their hands for asking so many doubts. Still, they may have some doubts, and that can be cleared when you come for theory classes or practical classes.
Yes.
And your lecture, really, it is enriching our students. And thank you very much. And for the students, I am reminding, tomorrow we have another lecture from Professor Raghuramulu Garu, and he'll be delivering on antigen-antibody reactions, immunology. You should not miss it, and this would be our last lecture in this week. Today's strength is also very bleak, just touching only 40. I expect more and more students in the tomorrow's meeting. Thank you,
Yeah, thank you.
once again.
Yeah,
Thank you very much.
Thanks for giving me this opportunity. So if you have any doubts, you can ask me in the class. Okay. Yeah, thank you.
Okay, thank you very much.
Yeah, thank you.
So all of you, thank you for your cooperation. And tomorrow also we have webinar, one more webinar. All of you attend tomorrow webinar. Thank you all.
Idiot, what are you doing? Hey, idiot. Hey, pa. Behave like a man. Okay. Shush. Ma. Video పోయిందన్నా. వీడియో నేను కూడా పోయాను. ఓ అంగిస్తుంది. అంగొచ్చు. వీడియో బాగుంది.
ఏమైపోయింది దేంట్లో ఉంది అప్పది జూమ్ ఏది
ఇప్పుడే డౌన్లోడ్ చేసిన
అవసరం ఏమంత ఇన్స్టాల్ చేస్తారు